Thank
you, Marie. I'll just share my screen. Okay, hopefully that's visible now for everybody. So yes, thank you very much, Marie, for the introduction, and thank you very much to safefood for the invitation to speak to you all today. My name is Aoife Colgan. I'm a senior epidemiologist on the gastroenteric and zoonotic pathogens team in the HSE Health Protection Surveillance Centre. And today I'm going to provide an update on the epidemiology of Salmonella surveillance in Ireland over the last 20 or so years, and then provide more detailed focus on recent cases and outbreaks. So for those of you who don't know us, we're the HSE Health Protection Surveillance Centre, HPSC. We're Ireland's specialist agency for the surveillance of communicable diseases, and our mission statement is that we strive to improve the health of the Irish population through provision of the best possible information on infectious diseases through surveillance and independent advice, epidemiological investigation, research and training. Our offices are based on Middle Gardiner Street, and if you're interested in following us on any of the social media platforms, we're on Twitter, Instagram or LinkedIn, so please do give a follow if you're interested in any of our outputs. So I'm just going to start with a brief description of the notification of infectious diseases process in Ireland. At present, there are 89 notifiable diseases in Ireland, and under the Health Acts of 1947 and 1953 and the Infectious Diseases Regulations of 1981 and then the subsequent amendments of those regulations, all medical practitioners, all clinicians and medical practitioners, including microbiologists and clinical directors of diagnostic labs, are required to notify to the medical officer of health, which is typically the director of public health in regional public health areas, any cases of statutorily notifiable infectious diseases or any unusual clusters or changing patterns of illness. The MOH, Medical Officer of Health, or the MOH, then has the responsibility and authority to investigate these notifiable disease cases and outbreaks. And they also have a requirement to notify to us in HPSC. HPSC then has the remit to conduct surveillance at a national level. We also provide national and international operational support, training, research, policy advice and public information. And we also are required to report any infectious diseases information to Europe through liaising with ECDC or the Early Warning and Response System, or more globally to the World Health Organization and relevant health authorities in individual countries in compliance with the international health regulations. So on the last slide, you may have noticed this little logo that says CIDR. So I'll go into a little bit more detail about what CIDR is. So it's our computerized infectious diseases reporting system, and it's a secure online national database for the notification and surveillance of infectious diseases in Ireland, complies with national data protection legislation. And CIDR is housed in HPSC, but it's a shared national information system for our CIDR partners, including regional public health areas, the Food Safety Authority, safefood, and the Department of Health. So just a bit of description of where CIDR fits into the ID notification pathway. So clinicians can make clinical notifications to the MOH. These can come via email, phone, post, fax, but laboratory notifications come from public health labs to the directors of public health via the online notification system CIDR. And then within regional departments of public health, the clinical and laboratory notifications are combined to make either events or outbreaks on CIDR, and these are notified to HPSC. HPSC can then use CIDR to generate outbreaks to assist in its functions of surveillance, op support, training, research, policy advice, and providing public health information. So I'll get straight into to describing the epi of Salmonella in Ireland over the last 20 or so years. So just focusing on non-typhoidal salmonellosis at the moment, between 173 and 456 cases of non-typhoidal salmonellosis have been notified annually between 2002 and up to the end of 2022. So this is a crude incidence rate of between 3.6 and 11.5 per 100,000 of the population. What we've been seeing generally is a sort of general decreasing trend in Salmonella notifications since the early 2000s. 2020 and 2021 obviously saw quite a drop off in the notification rate, likely due to effects of the COVID-19 pandemic. But the notification rate in 2022 has increased again to what we were seeing at pre-pandemic levels. So there were 342 cases of salmonellosis notified in 2022, giving a crude incidence rate of 7.2 per 100,000 of the population. Looking at the breakdown of cases by sex, we see typically an even split, males to females in our cases. So since 2002, 49% of the cases have been females and 51% of the cases have been males. And looking at 2022 specifically, the highest sex-specific rate was among females aged 0 to 4 years, with a notification rate of 23.5 per 100,000 of the population, followed by males also aged 0 to 4 years and a notification rate of 20 per 100,000 of the population. So looking a little bit more detail at the age distribution since 2002. The highest age-specific notification rate has been among children in this graph, represented by the red line. This shows 0 to 9-year-olds, but as we saw on the previous slide, the highest burden of disease is typically in the under 5s. Similar to the overall notification rate, the notification rate among children was generally decreasing since the early 2000s. Dropped off during the pandemic years of 2020 and 2021, but the notification rate among children in 2022 is at the highest rate that's been seen since 2012. After children, the group next most affected are the 20 to 29-year-olds, and this is likely due to proportions of foreign travel and perhaps socializing and dining out among this young adult age group. Looking at the geographical distribution of our cases since 2002, we typically see very little variation in crude incidence rate between the different HSE areas. In 2022, the highest, the crude incidence rate for each area varied between 5 to 8.3 per 100,000 of the population, and the highest crude incidence rate at 8.3 per 100,000 was in the northeast, and this is an area that covers Louth, Meath, Cavan and Monaghan. So we know there's a seasonal pattern of salmonellosis notifications in Ireland. The highest notification rates are typically seen during the summer months, coinciding with increased foreign travel and perhaps differing dining habits. There was an interruption to the seasonal pattern of salmonellosis notifications during the COVID-19 pandemic, obviously due to restrictions on foreign travel and socializing. And the seasonal pattern hasn't really recovered even in 2022, despite increased foreign travel and lifting of restrictions on dining out. So it'll be interesting to see how things progress in 2023. Just looking at the average count of salmonellosis notifications per month between 2002 and 2022, so that's here in the red line on this graph on the right, we can see that despite variation year to year, typically the peak of salmonellosis notifications occurs in August or September. Okay, so we've collected data on patient type and hospitalization status of patients since 2010. And since 2010, between 30 and 44% of notified cases of non-typhoidal salmonellosis cases were hospitalized. An additional 4 to 19% accessed emergency hospital care. But typically each year, the majority of patients attended their GP. So we would see proportion of patients attending GP between 36 and 45% since 2002. The exception to this, I suppose, was in 2020 and 2021, again, the pandemic years, because milder salmonellosis cases were probably less likely to be diagnosed due to limited access to GP care during that time. So what we were seeing was a smaller number of cases and a higher proportion of severe cases because these were the ones being diagnosed and being seen in hospital. We have information on symptom status reported on cases since 2012. And since that time, between 95 and 100% of cases were symptomatic. Diarrhea followed by vomiting and then abdominal pain were the most common symptoms reported amongst cases. So all Salmonella isolates isolated in primary labs are typically referred to the National Salmonella, Shigella and Listeria Reference Lab which is based in Galway. And in the reference lab, they perform they confirm the Salmonella isolates and perform subtyping and also whole genome sequencing. So we have this typing data available on CIDR, and we see that Salmonella enteritidis and Salmonella typhimurium are the most common non-typhoidal Salmonella serotypes in Ireland. The other most common serotypes that are seen are Newport, Infantis, Stanley, Burkow, Kentucky, and Agona. And in 2022, 22% of the cases were caused by Salmonella enteritidis and 33% by Salmonella typhimurium, including monophasic Salmonella typhimurium. And 38% of the remaining cases were either other or untyped Salmonella serotypes. Okay, so I should have mentioned earlier that we typically collect a core data set on all notifiable diseases in CIDR. So this includes age, date of birth, county of residence for the case. But for some diseases, and Salmonella is one of these, we also collect enhanced epidemiological information that allows ourselves and our colleagues in public health to understand a little bit more about disease progression in the country. So some enhanced data that we collect on Salmonella includes information about patient recent international travel history and potential exposures. So country of infection was reported for 85% of salmonellosis cases notified since 2010. And where country of infection information was available to us, we know that 57% of the cases were domestically acquired, so the country of infection was in Ireland, while 43% were associated with foreign travel. And when we look in a little bit more detail at those cases that were associated with foreign travel, we can see that earlier, between 2010 and 2015, the majority of travel-associated cases were to countries outside of Europe, whereas from 2016 onwards, with a few exceptions year to year, the majority of our travel-associated cases have actually been within Europe. The 5 most commonly reported countries among our cases associated with foreign travel were Spain, Thailand, India, Poland, and Turkey. And where international travel status was known, we see that there was typically that Salmonella enteritidis was typically more associated with travel cases, and typhimurium was more associated with domestic cases. So Salmonella typhimurium accounted for 41% of domestic cases and only 13% of travel-associated cases, while S. enteritidis accounted for 20% of domestic cases compared to 33% of travel-associated cases. We also collect information on whether cases dined out in the 3 days prior to their symptom onset. And this information is available for 72% of cases that have been notified since 2012. Where this information was available to us, we see that 64% of cases reported eating out in the 3 days prior to their symptom onset. And we also collect information on outbreaks or unusual or changing patterns of illness on CIDR. These data have been available for Salmonella outbreaks since 2004. And since that time, there have been 242 Salmonella outbreaks notified on CIDR. 64% of the outbreaks, the majority of outbreaks were family outbreaks as opposed to general or community outbreaks. And we see that the majority, 54%, have been associated with transmission in private houses or among extended family members. So they're generally quite small outbreaks. Other areas that we see Salmonella outbreaks are can be travel related or in hospitality or healthcare. Where information on the number of outbreak associated cases is available, outbreaks ranged in size from 2 cases to 71 cases. But 88% of the outbreaks notified on CIDR involved fewer than 10 cases, and we saw they're typically associated with household transmission. Where an outbreak extends beyond 2 HSE areas, this is typically defined as a national outbreak, and HPSC will lead that outbreak investigation. We also lead on outbreak investigations where there's a cross-border or international component. So HPSC have led the outbreak investigation for 14% of the outbreaks currently notified on CIDR, with a mixture of national and international outbreaks. Typical setup is that a multidisciplinary outbreak control team will be established, and this usually comprises colleagues from HPSC, the National Health Protection Service, the National Salmonella Reference Lab in Galway, our colleagues in the regional departments of public health, HSE Environmental Health Service, the Food Safety Authority of Ireland, and international agencies as necessary. Trawling forms are usually applied to cases in order to get a more detailed history, food consumption history and exposure history in the days prior to onset of symptoms. So a couple of the larger outbreaks that HPSC have been involved in leading the outbreak investigation for in Ireland in the last couple of years, I've just highlighted here. So there was one last year between January and April, an outbreak of Salmonella, monophasic Salmonella typhimurium in Ireland, but this was actually linked to a larger multi-country outbreak highlighted here in this ECDC and EFSA rapid outbreak assessment, the multi-country outbreak of monophasic Salmonella typhimurium linked to chocolate products. Another multi-country outbreak highlighted here that Ireland had cases involved in was a multi-country outbreak of Salmonella brandera, which was linked to imported melons. A large outbreak in Dublin in 2017, this was actually not led by, the outbreak investigation wasn't led by HPSC, it was led by HSE East, and HPSC provided operational support. And this was a very large outbreak associated with a catering company. There's also an outbreak associated with pilgrims travelling to Medjugorje, and an outbreak of Salmonella Heidelberg associated with in-flight catering in 2014. These are just some of the examples of the larger outbreaks that have been investigated over the last number of years. There are also smaller ones ongoing locally and nationally. Okay, so we also collaborate very closely with the National Salmonella, Shigella and Listeria Reference Lab, as I mentioned, in Galway. Most Salmonella isolates from primary labs are referred to the NSS-LRL for confirmation of subtyping. And at present, they conduct whole genome sequencing to predict antimicrobial sensitivity and for cluster detection. And cluster detection has really become a key aspect of our regional, national and international public health investigations over the last number of years, as it's led to the identification of new outbreaks, the confirmation of recognised outbreaks, the linkage of cases to outbreaks in other jurisdictions, such as those larger international outbreaks that I highlighted in the previous slide. The whole genome sequence helps us recognise the limits of the outbreak when an outbreak can be declared over, and also helps us to recognise when certain clones have become endemic. So there were 355 isolates with primary sample dates in 2022 referred to the NSS-LRL last year, and 43% of these were linked to new or previously identified Irish or international clusters, and the remainder were sporadic. As mentioned earlier, HPSC also liaises closely with our international partners, such as ECDC and other European or international organisations. In terms of the gastro diseases, we do this via the Food and Waterborne Diseases and Zoonosis Network, FWGNet. We also report annual Salmonella notification data through the European surveillance system, TESI. And HPSC and NSS-LRL share EPI and micro data through EpiPulse, which is an online reporting portal which enables early threat detection, monitoring, risk assessment and outbreak response at an international level. So public health authorities can report unusual cases or changing patterns of illness that they may see to other partners in the FWDNet. HPSC also contributes to international reporting of foodborne diseases through annual reporting of foodborne outbreak data to the World Health Organization and the European Food Safety Authority, EFSA. So this is just a screenshot from the ECDC Surveillance Atlas of Infectious Diseases. I just wanted to highlight the Irish notification rate in the context of the other European member states. So this is based on the data, the annual data that we report to TESI each year. And the most recent data in the Surveillance Atlas is up to 2021, which we know is an unusual year in terms of the fact that we had a lower notification rate, and most countries probably did. But actually what we can see here This line in blue represents the Irish notification rate since 2007, and the line in orange, hopefully you can see that, is the average of the EU or EEA notification rate since that time. And what we can see is, happily, our notification rate is significantly lower than the EU average. We can also see that represented on the graph, that our notification rate in 2021 was between 0 and 9.9, so we're on the lower end of the scale in terms of notification rates throughout Europe. And here on the bottom right, we can see breakdown of notification rates by age group again, Ireland in blue and the EU average in orange. And although our rates are much lower, we can see that in Europe, similar to our own picture, the highest burden of disease is in the 0 to 4-year-olds. Okay, so I'm coming towards the end of my presentation, but I just wanted to highlight so that all of the previous slides related to non-typhoidal salmonellosis, but we do also collect data on typhoid and paratyphoid in Ireland. These case numbers are much lower, but I'll just go through our case numbers and notification rates over the last number of years, just to highlight the picture for those diseases in Ireland. So for Salmonella typhi, we see between 2 and 31 cases per year since 2004, giving a crude incidence rate of 0.04 to 0.65 per 100,000 of the population. And with the exception of the pandemic years, what we have seen in the last number of years in 2019 and again in 2022 is quite an increase in S typhi cases. So they're shown here in the red bars and line. In terms of Salmonella paratyphi, we have a crude incidence rate of 0 to 0.22 per 100,000 of the population. So it's been between 0 and 10 cases per year since 2004. And cases of typhoid and paratyphoid are most frequently associated with international travel. Countries most frequently seen among our cases are India, Pakistan, and Bangladesh. Okay, so that's the end of my presentation. I just quickly run through some technical notes. So the data presented here are based on statutory notifications that were extracted from CIDR. The data are provisional and subject to ongoing review, validation, and update. And CIDR is a dynamic database that is constantly updated. So as a result, the figures reported here may differ from previously published figures or figures published in the future for the same time period. Population data were taken from census 2002, 2006, 2011, and 2016. And as mentioned previously, if you're interested in following any of the HPSC outputs, we are available on Twitter, Instagram, and LinkedIn. Finally, I'd just like to thank all of the data providers, so anyone anyone participating in the collection and reporting of data that were used in this report, including notifying clinicians, laboratory staff, public health doctors, nurses, surveillance staff, microbiologists, and administrative staff. I'd also like to thank my colleagues on the gastroenteric and zoonotic pathogens team in HPSC, Dr. Paul McKeown, Specialist in Public Health Medicine, Dr. Patricia Garvey, Principal Epidemiologist, and Dr. Catherine Timoney, Epidemiologist, as well as former GZB team members, Sarah Jackson and Fiona Cloak, and the COVID-19 business continuity team, who kept the gastro team show on the road during the pandemic. I'd finally like to thank SafeFood for the invitation to our team to present the up-to-date surveillance data on salmonellosis in Ireland. And finally, thank you to all for your attention. I'm happy to take any questions at the end during the Q&A, and I'll pass over to Frida for her talk now.
Thank you very much, Aifee. I shall get my screen up. Hopefully we can see my slides. Can you all see my slide?
Yep, perfect. Thank you,
Brilliant.
Frida.
Yeah. So thank you very much for this opportunity to share some lessons learned from some Salmonella outbreak investigations we've had in recent year. So I am working in the UK HSA Food, Water and Environmental Microbiology Services. We have 3 labs. I'm located at the one that's at Porton Down in the UK. And we get involved when we investigate outbreaks with Salmonella. And I wanted to have a bit of a lab perspective on this topic. I hope you'll forgive me. I'll try and see how we have used PCR in recent years to help us with investigating quite complex outbreaks. And amongst that, one with Typhimurium and Enteritidis from 2 completely different reservoirs, sheep and chicken. But I'll also include a little bit about a recent outbreak on melons, the one that Afi was mentioning, and some pork scratchings. And picking up with the burden associated with non-typhoidal salmonellas. So worldwide, it's causing over 90 million cases of gastroenteritis. In England, we have approximately 800 laboratory confirmed cases we report each year. Just to highlight that we know we under ascertain. So in reality, there's probably almost 5 times as many cases out there in the community. And there's a calculation that the burden of foodborne disease associated with non-typhoidal salmonellas was about just over £200 million annually, published in the UK report, the Foodborne Disease Report. Just to give a bit of background, we know from EU reports, so data from 2019, we're saying that almost half of the cases are associated with Salmonella enteritidis. The next most common, Salmonella typhimurium, and after that, infantis crops up at a much lower proportion, although the proportions of the different serovars associated with cases across the EU probably has changed in 2020 and 21, although the most recent data aren't available yet. In contrast, from chicken, the most common isolation reported is actually infantis, and enteritidis next most common with just under 8%. And just to give a bit of context in terms of the outbreaks I'll go on to talk about, there's considerably mandatory surveillance in place for Salmonella in poultry you'll be familiar with, but actually none for sheep. And in fact, sheep aren't really associated with frequent detections of Typhimurium enteritidis. For example, the far most common Salmonella to encounter in sheep is diarrhea zona. So I just want to introduce a little bit about the lab testing. So in recent years, we've taken advantage of screening samples using a PCR before we go on to do the whole Salmonella culture. So this helps us focus our efforts on the ones that really do have Salmonella in them. And on this one, you can see that this assay is able to detect Salmonella typhimurium enteritidis separately alongside all other Salmonellas. So you can see the green one coming up here means that we've got Salmonella typhimurium in the sample. And that is, of course, a part of Salmonella spur. But the blue one here is flat, suggesting that there's no Salmonella enteritidis in the sample. On this other sample here, we have enteritidis coming up. So, but no typhimurium, no green, and that can be quite helpful if you're trying to hunt down a particular serovar in your samples. So we used this the first time when we investigated an outbreak associated with sheep, starting in 2017, going into 2018. This was a new SNP type, which is what we use in the UK based on whole genome sequencing to type our Salmonellas. And we saw an increase in this particular strain. And we investigated this by looking at which abattoirs were affected, how an initial detection of sheep being unwell probably was the source of it. The Animal Plant Health Agency was helping farmers and traders and transporters to try and get on top of this. The Food Standards Agency were issuing warnings about ensuring consumers were alerted to cooking lamb properly. And there were a couple of particular raw lamb dishes that were highlighted as one of the contributors to the outbreak. From a lab perspective, you can be presented with a whole diverse range of samples. And I must admit, in this outbreak, I've never seen such quite scary samples as the ones we were coming across taken at the slaughterhouses. Nevertheless, our PCR helped us through them. So we tested over 900 samples from 14 slaughterhouses and used this PCR to screen to allow us to focus on detection of the presumptive Typhimurium samples. In fact, we had 8% of the samples that were presumptive positive, and we managed to confirm Typhimurium from all of them. But we also detected other Salmonellas in the samples, and most commonly diarrhea zona. But in fact, there were 5 other serovars also in the samples. And the rapid identification of the affected premises allowed more prompt intervention. So the food chain information follow-up and declumes in the slaughterhouses was part of that intervention. And as I say, the APHA were issuing guidance to help farmers and hauliers. The Food Standards Agency were following up with warning consumers to cook lamb dishes properly. There was detailed aspects of this, including better scrutiny of animals on arrival at the abattoir and checking paperwork on the food chain information, and doing deep cleans to try and eradicate Salmonella in the slaughterhouses so that there weren't remnants there. There were also more tracking of sheep movements, and there was risk assessment of on the farms. And the guidance given to the farmers hopefully helped them to get on top of this problem. We certainly have seen a much lower rate of this in recent years. There was a warm meats procedure that had been allowed for a bit, but that was discontinued. So I now want to talk about a Salmonella enteritidis investigation, where we in 2020 saw a rise in a particular type of enteritidis. And it was published in this rapid outbreak assessment you can see on the right-hand side here. And we investigated this particular type of enteritidis alongside some other enteritidis clusters. In fact, there were 3 other clusters we investigated at the same time, as they appeared to come up with the same risk factor, i.e. associated with exposure to raw frozen breaded chicken products. So across Europe, there were actually cases associated with this particular strain. You can see here with the UK in beige being the most prominent proportion of the cases in the outbreak. Our involvement in it started in 2020, where a remnant of some chicken nuggets were taken by an EHO from a case home, and we got it into the lab and checked it out with our PCR. And we could see that there was plenty of Salmonella there. So the red line comes up early. That means there's plenty of Salmonella there. The blue line is the Salmonella enteritidis. So we knew the sample had Salmonella enteritidis in it. Unfortunately, it's coming up really late compared to the red one. That means that there's a lot less enteritidis in there than there are other Salmonellas. And on a plate, you can't tell the difference between Salmonella enteritidis, Typhimurium or something else. And when we sent it off to our reference laboratory, they were telling us that the strain we had isolated was Salmonella infantis, which was a bit sad considering we knew there was some enteritidis in there, and we were trying to identify the Salmonella enteritidis outbreak strain. So we used a very old-fashioned method to try and get the Salmonella enteritidis out. There is something called a Craigie tube, which is basically a semi-solid agar with a hollow glass tube in the middle. So I was trying to use a pen, never mind. So what we did was take our mixed enrichment broth of Salmonella. So we knew there was Salmonella enteritidis in there, and we also knew there was other Salmonellas in there. We put it in the middle of the hollow tube, and the semi-solid agar contains antibodies against flagella to the infantis that was in there as well. So that means the infantis cannot move through the tube, and only the enteritidis can move through the tube and up to the other side, where after incubation overnight, we were fishing it out and culturing it onto plates. We took the colonies, PCR'd them, and realized that we had actually isolated a Salmonella enteritidis from the sample. And that came back from the reference laboratory as, in fact, indeed being the outbreak strain. So much happier. On the back of that, we decided to see if we could find other products on the market. So we did a quick reactive survey and took lots of details on them. And in fact, by doing this reactive survey, we identified 5 further products affected by Salmonella. So one product shown here, in fact, we detected 3 different clusters of enteritidis in the product alongside infantis and Newport. There was another product with enteritidis and infantis only, also another one with infantis and enteritidis, and finally a product with only enteritidis, alongside the original product, where we ended up having all 3 different SNP clusters of the Salmonella enteritidis detected alongside some Infantis and Newport. We tested only 4 packs of this product, and all 4 packs were positive. So these were on a regular basis then included in the RASFF notifications, and the producers were informed by the Food Standards Agency to try and get rid of them on the market. I just wanted to highlight that we came across several samples, like the first one, that had a mixture of different salmonellas in there. So you can see these 5 products here. And for example, product B, we had 1 sample with Infantis and then 2 other types of Enterovitis in them, alongside another sample with Infantis and just 1 of those types isolated. So you can see it can become quite complex when you are trying to hunt down a particular outbreak strain, if there are other strains in there that may interfere with your ability to detect them. And on a plate, as I say, you can't tell the difference. And some of these samples were extremely difficult to isolate Salmonella from. So the lower part of the plate, for example, here, giving us hardly any growth of Salmonella in the RBS broth. When we were looking at the phylogeny of the strains, we were realizing that on this tree here, you can see that the original outbreak strain we were trying to hunt down, the 4833, is quite distant from the main cluster T2512, with 2 subclusters, T512 and T590. And what's so brilliant about the whole genome sequencing is that it can give you really quite a detailed look at the genes in the Salmonella strains. And what we were able to establish is that one particular origin plan, PL1, seemed to have a slightly different variety of the T5590 strains compared to the other plant PL2 in Poland, that had a slightly different type of the T590 present. Whereas the T512 we were only able to associate with PL2. So the outcomes were published in a scientific paper, and there was quite a lot of press attention about it. But adding on this survey enabled a wider source finding, considering the majority of these positive products have really quite a long shelf life. That was really important, and the PCR helped us focus on isolation in the samples that were affected. So we recorded something like 54 production plants, but only 6 of them we ever had any Salmonellas associated with. And in fact, looking at where those plants, some of those plants had imported their meat from, we tracked all of the positive Salmonella products back to plants in Poland. And while we started with one particular outbreak strain, we also managed to find other Enterovitis outbreak strains in the samples. Intriguingly, while Enterovitis was causing the outbreak, we actually more commonly came across Infantis in the products, possibly saying something about the relative either level in the products or in virulence associated with them. And just to highlight this issue with co-contaminated samples, where you may have an Infantis that masking the detection of an outbreak strain if it's much more numerous in the sample. And there was plenty of public communication to advise the consumers to ensure that they really were cooking these products. And as I said, plenty of withdrawals and recalls associated with this action. And I want to say a little bit about a melon outbreak. I'm really sorry, I think I'm beginning to run out of time, but I will try and finish on time. So Galia melon, as was mentioned earlier, was associated with an outbreak of Salmonella Brennerup, and you can see the rapid outbreak assessment on the right-hand side here. We did a case control study and got the biggest odds ratio I think we've ever seen, 672. And we were able to get hold of some melons from an importer, and we thought we would try and have a look at these melons. So we were given 200 melons, and we were quite careful to get the supplier details absolutely nailed on these. And again, we used the PCR screening of these 200 individual melons, testing the rinse of the whole melon alongside rindens in the same buffer peptone water enrichment broth. Previously, there had been 2 other isolations, although none of those, one in Finland and one in Austria, weren't able to have sufficient traceback information associated with them to prove where the original contamination was coming from. So you can see the Galia melon here, and tested 200 of them, and these were the boxes they came in, and this little almost anonymous sticker here was in fact the information you needed to be able to know the exact origin of the grower of these melons in Honduras. So the outbreak strain came back reported, and in the ROA you can see that the investigation here presented that the 200 melons were tested, and 2 of them were positive. So perhaps we were lucky then. And yes, the outbreak strain was confirmed and traced back to a grower in Honduras that was investigated by the authorities there, as well as the FSA in this country. Lastly, I just want to say about an outbreak we had associated with pork scratchings, perhaps not the first kind of vehicle you would associate it with Salmonella. I mean, they are fried and often triple fried. But yes, we had an outbreak in 2021 that was widely published, and we were not initially very convinced about the epidemiological signals, but it just goes to show how careful you have to be on getting the epidemiological information, and the signal to suggest pork scratchings turned out, in fact, to be correct. So despite the disbelief that Salmonella could be present on a product like that, that was established. And again, we undertook a reactive survey. So in addition to the original products that were withdrawn, we also found a completely different supplier and a completely different Salmonella in some packs of pork scratchings, and they were also eventually withdrawn. So again, a survey to follow up on whether there was wider contamination present in such products showed that we were able to identify further products to be withdrawn, and there were also cases associated with that Salmonella. So I'll just end with acknowledging people within UKHSA and the FWE labs in particular, all our Salmonella surveillance specialists, including Leslie, Annie, Jackie, our reference laboratory in Collindale, our epidemiological colleagues, and people from our bioinformatics department, alongside the FSA and the local environmental health teams for helping with the food tracing and enforcement and sampling. So thank you very much, and I'm sure there'll be questions in the Q&A session following this. So now I want to introduce Gary and Mahon, who is going to talk to us about Salmonella in the industry, I believe.
Thanks, Frida. Share my screen. Hopefully you can all see that now.
Oh, good. Thanks, Gary.
Okay, thank you. So yes, as Frida mentioned, I'll be speaking about the sort of controls we have in the poultry industry, particularly within Moy Park itself. So just to give a bit of background to those people who don't know who Moy Park are, we're one of the largest, part of the Pilgrim's processors of Pilgrim's Pride from the US. We're one of the largest chicken producers in the world. We were set up in 1943, and after a management buyout in the 1980s, were then acquired by the OSI group, and then further acquisitions of GB businesses, which included Padley's and Dove Valley, and then the Northern Ireland business of O'Kane's, were then purchased by JBS, and then finally by Pilgrims, as a part of the largest company in the world. Our sort of structure is quite unique. We've got a young farm estate. We've got facilities capable of supplying next generation. We've got a unique performance house, which allows us to do trials, looking at 18,000 birds, feed formulations, alternative breeds and bedding. We've got a strong European geographic biosecurity because we're based in Northern Ireland. We've also got our brand new hatchery in Newark itself as well. We also have quite a complex supply chain, moving through from feed mills, grandparents, parents, broilers, through to our processing sites, which are fresh poultry or prepared food sites as well. So again, from a laboratory aspect, we have 3 laboratories within the UK. We have 2 in Northern Ireland at Ballymena and Craigavon at our headquarters site, and we also have 1 in Alnwick in Lincolnshire in England. Just give you a bit of an idea of the structure we have to have in place to maintain these laboratories. So I have myself as company microbiologist. We have Pamela Mullen, who's our group laboratory manager, and then we have our 3 laboratory managers looking after the 3 separate laboratories. And under them, then we have senior analysts and analysts. Plus, as part of the sort of group support team, we have our LIMS manager, our quality compliance manager, and then our laboratories coordinator as well. So we have a team of about 30 across 3 laboratories and locations, including those central functions. We're all accredited laboratories. We have UCAS for regulatory salmonella analysis and CLAS for all of the analysis. And we're also, all 3 labs have got RSA Gold for pathogens. So we use all our testing for salmonella is done in-house. We currently spend roughly about 1.25 million on our internal analysis spend, and that covers about 100,000 total samples analysed. Of those 144,000 samples, 53 of those are actually salmonella analysis for agri, and 10,000 are for salmonella analysis for food. We do use traditional and rapid analysis, which I'll come on to later. I'd also like to point out we've also spent over £1 million and opened a brand new lab in our Alnwick facility in Lincolnshire. Just a few photographs here to show what we've done. So that this has increased our capacity and our footprint quite significantly within our GB sort of testing facilities. Also, it brings brand new facilities for our colleagues there as well. So I'm not spend too long on this, as obviously Fred has touched on it as well. But sort of the sort of list of the top servers being reporting about reports. I just want to highlight that some of these are actually regulated for poultry. So we're into the Typhon Miriam, obviously, for our broiler farms, but also as well as those 2 for our breeder farms, we also have Infantis and Verca. And you can see how these sort of stack up within the sort of the top list there. Also looking at the foodborne outbreaks from 2019 on Typhoidal, we have Indiana come through there as the one that was identified as linked to chicken. So within the sort of national flock, the regulated servers have sort of remained very low for SE and ST. However, other non-regulated servers have shown an increase in recent years. So 3%, 3.5%, 3.7% up from 2019 to 2021. And 2022 should be published in Q3, Q4 of this year. As also touched on by Frida, then Salmonella infantis, whilst regulated for breeders, it's not for broilers, but it's also given concern then within broilers as well, and that's been considered. This is some of the regulations that we have to adhere to, so I'm not going to go through these in any detail, but it covers everything from breeders and broilers, hatcheries, factories and sales testing, and also any byproducts too. So I'll sort of split this presentation then into agriculture division, and then I'm moving on to the factories. So to start off from agriculture division, just give an idea of our sort of spread of farms and factories and hatcheries, etc., and mills. This is where we're sort of clustered around in Northern Ireland, and you can probably see there's a cluster in the southwest of Northern Ireland and also a cluster in the northeast. So southwest is traditionally where was the original White Park farms, etc., and also in the northeast is where originally was O'Kane's, which is now obviously part of the White Park group over the past decade or so. Then within GB, you can see it's probably spread out a bit more, and this is due to obviously our original sites here were through Dove Valley, which is based around Ashburn in Derbyshire, and then the Padleys group, which was based in Lincolnshire and East Anglia. So a bit more spread, a bit more area to spread out into for broiler farms and your farms as well. This slide here just sort of shows, this slide will be repeated several times as I build on it. So on the left-hand side there, you can see going from pedigrees to great-grandparents, grandparents, parents, and broilers. So the different generations of actually the supply chain of the birds themselves. Obviously, ourselves in White Park, we're quite unique in that we have the grandparents, the GP, and the parents, and the broilers within our control. And then our sort of pedigrees are within our breeding house. For example, Aviagen, that would be in the pedigree and the great-grandparents. The other thing to note from that left-hand side there is that if you want to make any sort of changes to the bird, its characteristics, whatever, you sort of implement those at the pedigree stage. So it takes 4 years, as you're seeing, to move from that changes you're trying to implement in the pedigree stage for it to actually come down to the broilers, which are the birds that end up on your table. And also you notice the numbers increase as well. So it's a bit of a pyramid where you have 1 male, 10 females at the pedigree stage. That ends up being over 48 million broilers by the time that works its way through. So within the sort of structure and supply chain, there are lots of controls there as well. So look, we start off looking at feed, looking at the raw material supply chain, having approved mills, having testing there as well. Within breeders, we have designated biosecure farms, restricted access and testing, biosecurity around transport, hatchery to rearing, rearing to line, and eggs to hatchery. Hatchery itself is basically like a high-risk factory. We've got a biosecurity, restricted access, testing, internal design barriers. Within bedding, which goes into the farms, we've got raw material supply chain. We have approved suppliers and testing as well, which goes on. Water, we have secure supply and treatment, and also testing that water. And so any inputs coming into the farm at all, they're tested and through a supply chain, secure supply chain. And in the farm itself, the biosecure design, restricted access, so intercrop hygiene, so in between flocks, approved in a lot of the hygiene team as well. And then the day-olds which come from the hatchery to the broiler farms, there's biosecurity around the transport to the farm as well, the hygiene around introduction of bedding, feed, say day-olds, and then the dispatch to the factory. So biosecurity around the catch and collect transport, and then the hygiene, the modules and crates which are cleaned at the factory before they return out to another farm. So looking at some of the controls and testing and feed. So if we start off on the sort of top red box there, the raw materials from approved audit suppliers. So we do have a testing regime for raw materials coming in, and actually it's the finished product as well. So any sort of raw materials coming from around the world, they'll be tested at the entry port. They'll be also tested by our sales, and then that will follow on. So we then sort of use approved hauliers. We have segregated fleets for raw materials and finished product, and we'll have a dedicated swabbing program as well. Raw materials added as per formulation. We'll also have biosecurity down to the facility, and then have a comprehensive swabbing plan. Same with the grinding and mixing stage as well. Then we get on to the next really important bit, and the bit for one of the controls around Salmonella is either the product can be then heated traditionally using kettles and ripeners, and but during that case we have to use employer higher time temperature. We have a validated programs for that. Or we can heat through long-term conditioners, which ensures first in, first out, and allows a lower time temperature validated program. So obviously there's energy consumption, but there's also a nutritional benefit from doing the slower, lower temperature. The same pressed into pellets are produced as a mash. We should, again, depending on the final use, the age of the bird, the type of bird it's going to, and then that'll be part of a swabbing program. The cooler then as well, there's a series of filters from 4 micron to 1 micron, and that's drawing the fresh air in from the outside. And then there's a coating of fats and oils and vitamins, and obviously vitamins, etc., are added at this stage because they're destroyed during the heating process. So there's a risk there. So again, that'll have to be part of a risk assessment and a control mechanism as well. And then it's sent to a sealed finished product bin awaiting dispatch to farm in dedicated vehicles. And then there's a dedicated swabbing program for those vehicles when they go out the farm, when they return from farms, etc., they'll be swabbed. And on the left-hand side, there's a time temperature and heat treatment ranges from either 80 degrees for 4 minutes or 86 for 6 minutes. And we also really consider Salmonella Seftenberg, which is a reservoir of concern in feed due to its very high heat resistance. So again, we look at the feed mills coming in, you can see the different spots in that sort of supply chain where they're coming through. And the end result, we're testing 2 by 25 grams per batch, and that batch can typically be anywhere between 3 tons to 5 tons. As we move on through then into breeder farms, so that aspect of time, we'll move through these quite quickly. But the one thing I probably should have said about the breeders is that the grandparent breeders, we only have them in our Northern Ireland base, and that's for geographical location to make sure, sort of keep them sensitive, keep them away from any sort of AI flows of birds, etc., keep them as remote as possible within Northern Ireland. So our GPs, our grandparents, are only within our Northern Ireland base, where we have parents or breeders within Northern Ireland and GB as well. So I'll not spend too much time flicking through this because we already touched on it, but I say it's just really biosecurity of the breeders, looking at the intercrop hygiene as well, biosecurity to the hatchery, the bedding coming in, and water supplies, etc. as well. But within the breeding, there's 5 regulated reservoirs. There's Enteritidis, there's Typhimurium, there's Hadar, there's Infantis, and there's Verca. So again, you can see the different tests here as well. Coming through, so we have composite feces, your breech swabs, etc. So you'll do these tests up the pre-point of lay, 2 pairs of breech swabs in, composite feces. So what we have, the birds will stay in the rearing farm until they're probably about 19, 20 weeks of age, until they become mature. Then they are transported across to a grandparent breeding and laying farm. So that's farms specifically designed to collect eggs. So they'll move across to that. And here they'll spend probably up to about 60 or 65 weeks of age in total. And again, there's a collection of different tests we do at that point as well, and carry out at routine intervals through at the point in time. And then the birds are then sent through, the chicks or the eggs are then sent through to the hatchery, the parent hatchery. And again, there's lots of checks done there within fluff, hatchery debris samples, eggs, shells, etc., tested as well. And environmental swabbing will go on. And then when the live chicks, after they hatch, they're then moved into the parent rearing collection. This is normally where other sort of poultry suppliers will come on board. We have these 3 extra steps to start before that. So here you look at box liners, basically where the chicks are in the boxes before they go out. The day-old, any mortality as well, we'll check those. And then every 4 weeks, we have 2 pairs of boot swabs or composite feces. And again, before we move those into the line farm, similar to the grandparent farm, we'll test them again. And then once they get into the breeding line, we'll have again another up to 60, 65 weeks. And every 2 to 3 weeks, we'll do 5 pairs of boot swabs or 1 pair of boot swabs plus dust swab as well, or composite feces, depending on the farm and what we're trying to do at the time. And then once the eggs are laid on the parent line farm, they'll then be removed and brought to the hatchery, which is the broiler hatchery. And again, we'll test here. So test day-old meconium, we'll do day-old mortalities as well, any sort of shells, etc., fluff and hatchery debris samples, plus any environmental swabbing we do, and plus any investigation samples we'll do as well. Okay, and we move on then to the controls and testing in broiler farms. So again, as I mentioned earlier, sort of the bedding, we'll check the water again, the farm, the day-old, something similar to what we did when we moved between all the different farms within the grandparent rearing and laying and the parent rearing and laying as well. The difference here then obviously was when the day-olds are sort of born and are hatched in the hatchery here, they then move not to a farm, they move to the they move to the broiler farm, and then once the birds from there, they move then to the dispatched to the factory, obviously where they're processed. These are the birds in that are part of the table birds as well. So again, important bit for the factory here is in is actually the cleaning and hygiene around the modules and crates of the factory before they return to farm, so to break any sort of cycles of contamination. There are 2 regulated cerevars here. There's the Salmonella enteritidis and the Salmonella typhimurium, and that includes monophasic Salmonella typhimurium as well. So again, reduction from 5 to 2, but there is discussions regarding potentially maybe increasing the regulation of cerevars here as well. So some of the controls we have on farm, I thought I'd give you some examples of that. Now some of these actually arose through other sort of project streams. So for example, traditionally, historically within the farm, you would have a line on the floor to designate between the clean and the sort of dirty area of the farm coming in. We've now moved away to that and actually have a system that is very akin to a high-risk factory. And that all came through as part of the Campylobacter sort of project. And then, as you can see here from the diagrams, we've got the color coded the areas. So outside the house is basically outside in the yard before you walk through the door into the house. So you'll dip your boots, you'll come in your outside boots, you'll leave your boots there, you'll swing out of those boots without touching the red floor and swing into the amber or yellow floor. There in your sock soles, you'll wash your hands, and then what you'll do, you'll swing out, and without touching the green floor, you'll swing into your boots for the house in the green area as well, and then you put on your coat and you sanitize your hands as well. So that allows for a clean break between the outside of the building and the inside. When it was only a line on the floor, it was very difficult to manage and very difficult to control that people didn't accidentally step over across that line. Now we have physical barriers in place that helps people to manage that and helps people to maintain that sort of biosecurity break between the outside and the inside as well. And obviously you do the reverse then on the return. That, I say, that was initiated by ourselves in Moy Park and is now part of the sort of the red tractor standard. We also then have the auditing as well. So every crop, every farm goes through an audit on biosecurity. Again, this was brought in as part of Campylobacter, but it has benefits obviously for Salmonella and for AI, etc. And it's a simple 1-page sheet. It covers all the basics from biosecurity signage to chemical storage, and has a RAG score then based on the overall audit. So every single flock, every single farm will have that RAG score. And that's the biosecurity audit was instrumental in helping to implement our Moy Park double barrier. So again, we started to introduce the double barrier at the start. there's something new for the farmers to work with. So we help that through the audit and through support. We help the farmers then move across this new system. So then we're on the broiler farm. Within 3 weeks of slaughter, we have 2 pairs of breech swabs, and this comes into 1 sample. At this point, if we don't, for whatever reason, don't process the birds within 3 weeks of that initial test, we will then have to retest again. So, for example, if for some reason the bird waits, we needed to keep the birds longer than the 3 weeks between the initial test, we'd have to go back and retest again before those birds can be processed on in the factory. There's also possible interventions at that point. If you can use organic acid in the water to achieve a pH around 3.7 to 4. However, even if it's used during the 3 weeks post the original sample and it delivers a subsequent negative breech swab result, the original flux status is still unchanged. So even if we put something in place to try and improve the Salmonella status of the birds, we still have to go by the original test. This sort of gives an outline just of all the sort of tests to date until the point of the broiler farm itself, all within agriculture. As you can see, there's quite a lot of sampling, quite a lot of detail around this that goes in place. And then within operations, there's controls and testing at the slaughter as well. So the legislation we need to have is this food chain information documentation. So all agricultural traceability information, so that's veterinary treatments, withdrawal periods, and the Salmonella status determined from the original breech swabs taken within the 3 weeks of slaughter, must be included on this FCI documentation. Which looks something like this. The FCI must be presented to site OVS in advance of the birds arriving at the slaughter site. The site OVS must then review and sign the FCI documentation. And then once the birds are processed, the Salmonella testing is then carried out in accordance with the Macrocriteria Regulation 2073. So again, as part of that 2073, then we're taking 5 samples per batch per product category. Just the next few slides will show sort of amount of product categories that we, as Moy Park, have to follow, ensure the sort of the safety of our birds. So again, the first one here is the food safety criteria under 2073. And the fresh poultry meat is the first category we come across. This is basically your primary chicken. This is your chicken with no added value whatsoever to it. So your plain whole bird, your plain drumsticks, your plain fillets, etc. Here you have the 2 regulated cerevars, the typhimurium and the enteritidis. These are as part of your food safety criteria. And again, obviously the criteria is not detected in 25 grams. So these are neck skins that are taken at the point X chill. And these are sampled then to determine the status for the Macrocriteria regulations. We then move on. So if you start to manipulate and cut that meat, you then have meat preparations and potentially minced meat as well. So the actual organisms of concern are not just SC and ST, they actually are all Salmonella species. So the criteria gets tighter in that respect. And I said, no, I'm still not detected in 25 grams across your 5 samples. If we produce MSM, it's slightly different. It's still all Salmonella species, but it's not detected in 10 grams, so a slightly less sensitive test. And then if you have many meat products made from poultry meat and tend to be eaten cooked, then it's back to something similar to the meat preparations and minced meat, not detected in 25 grams. And obviously, then we also have listeria if we do RT products as well as food safety criteria. Then we move on to the process hygiene criteria. Poultry carcasses or broilers. So we're in the food safety criteria, it was SC and ST. Within the PHC, it's actually all Salmonella species. Same amount of tests, and it's all, again, on neck skins. Also, at the same point in time, we carry Campylobacter tests as well as part of the process hygiene criteria. But as we move through, as we did before, meat preparations, you have E. coli as your process hygiene criteria. And then within minced meat, you have E. coli and also TBC as well. And it's a similar thing for MSM. And as I mentioned earlier, we also have Campylobacter, which is done at the same point in time as you're doing your testing for poultry meat and carcasses. So within our business, I mentioned earlier, we have different tests to be done. So within our Dungannon site, for example, we have primary, we have neck skins looking for Salmonella and Campylobacter, MSM, TBC and Salmonella, RSC, Salmonella, excuse me, primary added value, retail products. We have a range of meat preparations, and we'll have to cover those off across the different sort of HACCPs, et cetera, we have within the business, and that's Salmonella and E. coli. Obviously, Ashburn, something similar. We have primary and primary added value. And in Lincolnshire, we have something similar, again, primary added value. And also in Ballymena, within Northern Ireland, again, we've got a poultry meat, and we've got a premium added value as well. Grantham, which is one of our prepared food sites, which is coded in RTE. Obviously got meat preparation of meat products there. So again, Salmonella is part of that testing. And then also in Craigavon, something similar to Grantham. So we've got meat preparations and meat products, and there's Salmonella and E. coli testing there as well. So one of the quirks, as I mentioned earlier, looking at the sort of testing within the MEC criteria, but one of the quirks which doesn't really have a sense scientifically or from microbiology aspect is when you look at meat, poultry meat and meat preparations. So you look at these two carcasses photographs, they're both poultry meat in terms of the regulation. If you test and you find Salmonella enteritidis or Salmonella typhimurium, then that is a food safety criteria fail, and it's also a process hygiene criteria fail. If you find other Salmonella cerevars, that is a food safety criteria pass and as a process hygiene criteria fail. So in that respect, if you find SA and ST and your product is on the market, then you're in the recall situation. If you find other Salmonella species, then you may not be in a recall situation. The legislation doesn't require that, but you do require to obviously follow an investigation to understand where your contamination came from. However, if you add a simple piece of added value to that carcass, and that could just be as simple as a sprinkle of herbs, that poultry meat then becomes a meat preparation. Again, if you have SE and ST in that, that becomes a food safety criteria fail, and that becomes a process hygiene criteria fail. And obviously then that involves a recall where necessary. If you find other Salmonella species, in this case, then you also fail the food safety criteria as well as the process hygiene criteria. And this would also maybe necessitate a recall situation. So as you can see, the addition of a simple herb to our product, you suddenly change the whole status of the product itself. So we look at the sort of testing, sort of coming to and now looking at the different testing within our business is quite extensive. So just flicking through that, you can see the amount of controls we have in place across the business, across our supply chain. And then we also look to the future. So for that amount of testing, we always look at ways that we can try and improve. We try to speed up the testing that we do, and particularly as we have that within control within our own laboratories, we look to try and see how we can bring that sort of technology in-house. So if you look at ELISA technology, we do utilize that. We utilize that in our Craigavon laboratory, but it's only for foodstuffs. So where traditional methodologies, you may be taking 3 to 4 days, at least to maybe try and get a negative result. Within using this technology, we can have a negative result within 24 hours. That's quite good if you're trying to screen for a product, and particularly where you have a low levels of incidence or prevalence of Salmonella as it is. It's very good for doing this. Using this technology. However, when you want to go into other sort of matrices, such as potentially agriculture, where you've got a lot of background microflora, then there's issues. And when we've tried to trial this in the past, we haven't been able to successfully get that to work for agricultural type samples. Looking at lateral flow devices, we've looked at this particularly, well, not necessarily only for Salmonella, but looking for Listeria, looking for Campylobacter. Our problem with lateral flow devices has been the sensitivity. So, for example, when looking at Campylobacter, where you're trying to understand is your level below 1,000 CFU per gram, typically the lateral flow devices we have seen or been on the market are minimum level of detection or limit of detection is 10 to the 3. So it's not really beneficial in that respect. So lateral flow, if they improve, may become beneficial, but at this minute in time, we haven't found one that is successful. Also looking at PCR, as mentioned by Frida as well. We are currently running a trial on PCR. We've looked at PCR in the past for foodstuffs. It's never been as beneficial or competitive, particularly financially, compared to our ELISA technology. However, we're now looking at a trial on agricultural samples, and so far that trial has been running really well since last year, and we're getting really good results from that there. So we're getting really good sensitivity. We're not getting any sort of false positives coming through, etc. So again, we're hopefully being in a point in the very near future to try and present that, try and get that approved. But I said that's the way we want to go, because that will give us a result compared to our traditional ISO methodology a couple of days earlier. And then the other thing we obviously want to look at is whole genome sequencing as well, because within our business, obviously, because of the extensiveness of our supply chain, if we pick up a Salmonella at various points in that, which may not seem at first hand to be linked but actually from the actual typing we do, we realize there's similar strains. We want to go back and try and do more analysis to try and understand where the link is and try and do sort of look at the root cause of that as part of the supply chain analysis. At the minute, it's still fairly expensive, but prices are starting to come down, and hopefully at some point in the future. And again, that's why we built our new lab to try and understand, can we bring that sort of technology in-house, if not whole genome sequencing, maybe even next generation sequencing as well. What are the things we need to look at as part of this cost? It needs to be as cost effective as the current methodology. Unless there's a reason. If there's a huge benefit from new technology and it's more expensive, then we'll consider it. The limit of detection, as I mentioned, needs to be sensitive or better than the current method, needs to be able to deal with background microflora, needs to be able to mobility. Ideally, what we want to do is be able to try and do some tests on farm or in the factory, but that might not always be the case. Sample and product matrix, so make sure there's no interference or cross-reactivity. Look at the scope of tests and the regulatory approval, so covering the full range of required organisms. So there's no point having one technology that only tests for one organism. So ideally, if we done PCR, we wanted to test for Salmonella across agri, food, also Listeria potentially as well. So we'd wanted to include lots of other tests as well. Excuse me. Look at the speed, must be faster than the current traditional method. Specificity, looking at the level of false suspects and delaying getting final confirmation at cost, because we do with the current rapid technology, it's very good for screening and getting your not detected back. But if you get a suspect and then your initial confirmation doesn't give you the confirmation you need, you do have to go back again, and sometimes actually end up being slower than the traditional method. And then also looking at viable and non-viable. So it's important to know the result is not due to residue from historical issues. So if you have, for example, a Salmonella on farm and you put in a robust hygiene protocol, then if there's any sort of fragments of DNA about, you don't want a subsequent test and to pick up and say you've still got Salmonella on the farm, when in fact you haven't. There's no viable cells there at all. So that's something we need to consider. So that's all our considerations. And say that's my last slide. So I'll hand back to Maria on that. So any questions, I'll happy to answer some of the question and answers. Thank you.
Right, sorry, we can't actually hear you. You might just unplug your headset. That might be a bit of a problem on that.
Okay, can you hear me now?
Yeah, perfect. Yeah.
I'm mumbling away there, apologies. Headset failed. I was just saying thanks so much to Aoife, Frida and Gary for giving us 3 really brilliant presentations, really informative talks as to where we are in terms of Salmonella cases and outbreaks in Ireland and the UK, and indeed controlling Salmonella in the poultry industry. I see we have a lot of questions that have come in already, so if you have any last minute questions, pop them in our Q&A box at the bottom of the screen, and we can make use of all our experts' knowledge while we have them available to us. So some of the questions appear to be quite long, so you'll have to bear with me. Okay, so a question came in early on, so I assume that's directed towards you, Aoife. Someone's asked, was there any particular reason for the rise in Salmonella cases in 2022?
I suppose it's probably similar to what we're seeing with a lot of diseases post-pandemic. The lifting of restrictions on foreign travel and socialization means that maybe people are doing that more often now that they can. What we saw on that slide about the change in kind of the seasonal pattern, I think would suggest that there's probably travel happening outside of the normal periods now. And then people weren't exposed to pathogens for so long, so sometimes maybe are getting more severe infections than what they would have previously gotten when they're kind of constantly meeting pathogens over and over. So maybe there's more. And another thing as well, I think since the pandemic, there's more health-seeking behaviour. So any symptoms at all, people are more likely, I think, to go to a GP or to get tested. So it's kind of, I think, a combination of things. Yeah. And certainly what we've seen for 2023 so far for Q1 is that the numbers are still high compared to the same time period in previous years. So yeah, we'll be, I suppose, watch this space to see how it progresses for the year.
Yeah. Okay. Thank you, Aoife. Sorry, someone has asked also, and this was early on, so I think it's for you, Aoife, that they were asking, do you issue live alerts by means of, they're sent Twitter accounts, but I assume it's social media in general. So the HPSC on cases and outbreaks? if you're aware of this, just
Not
ask
so
them.
much, I suppose, kind of more reactive topics. For example, MPOX in the last year or so, we were probably issuing more reports more often, and that would be highlighted on our Twitter when a new report was available. I should have said also on our website, we produce a weekly notifiable diseases report, and so information on the year to date will be available weekly on our website, and that's highlighted on our social media channels when those reports are published. But in terms of cases and things like that, no, it wouldn't be sort of a live feed of anything like that. And for something like Salmonella, that's not as reactive unless there's a very large outbreak ongoing, we probably wouldn't have that on our Twitter. No.
Okay, thanks, Aoife. We have another question on, I think you mentioned it, Aoife. Actually, someone's asked, has whole genome sequencing replaced traditional methods for predicting antimicrobial sensitivity? I'm not sure who wants to take that one, but
I can say from the Irish perspective, it has in the reference lab. But if there was, we'll say, a very severely ill patient in hospital where antibiotic therapy was indicated for that person, I think the local lab in that hospital would have access to do antimicrobial susceptibility test, phenotypic testing in the traditional ways for clinically relevant antimicrobials that might be used for treatment of that patient. But in the reference lab, they have moved over to whole genome sequencing for prediction of antimicrobial resistance across a wider panel of microbials as well, I suppose.
Okay, thanks, Aoife. Frida, did you want to mention the UK?
So it's pretty much the same in UK HSA Ref Lab. All clinical isolates do get the whole genome sequencing and use that to predict the AMR profiles. But sometimes MIC testing is undertaken supporting, clinical decisions, like you say. For all the harmonized surveillance of Salmonella, mainly done by APHA, MIC testing is still done really to comply with the harmonized testing surveillance. Yeah.
Okay, thank you, Frida. Okay, someone's saying they work in a Salmonella testing lab and they've seen a significant increase in Salmonella infantis isolates from tested products. Given its increase has been noted in the EU, is there any discussion re-adding infantis to the list of notifiable strains for action in poultry? If anyone wants to take that question.
I mean, I've heard people talk about it, and certainly in the US, but I'd be interested in Gary's opinion on this. I think it's hard.
We've heard talk about it as well, but nothing definite as such. That's sort of hinted at in my presentation. Yes, there's talk about that coming in.
But there's almost more push in the US to actually, they might introduce a criteria including infantis earlier than the EU, potentially. I don't know. Might happen.
You could do, yeah.
Okay, perfect. I have a question for you, Frida. Both lamb, mince, and breaded frozen chicken outbreaks were, I suppose, products that are, considered or labeled with cooking instructions, so i.e. they're non-ready to eat. But poor consumer cooking practices seem to contribute towards consumers becoming ill. What is your opinion? Could public health agencies do anything more to raise awareness among consumers that these type of products, I suppose, are raw meat products and can contain pathogens and should be handled as such?
So in some of the outbreaks, our Food Standards Agency were in conversation with the producers to try and improve the labeling on the packs. I mean, sometimes the writing is so small you can't actually see how it's supposed to be cooked. And some of the heat treatments suggested were a bit marginal, we thought, as well. Another thing is, I don't know, celebrity chefs, just you could mention these things. But it is very, very hard to keep the consumer informed. And I still think the most effective point of communication is when they have the pack in their hand and they need to understand what to do. But in fact, with the breaded chicken products, we did realize that the companies had done some pre-pasteurization, if you like, of the product, and then to cut costs, that stopped for a bit. And that certainly increased the risks. And then that reveals that consumers probably regularly may undercook products, especially those that already look cooked, that some of these breaded things, they look cooked, so that's really hard. Thank you.
Okay, thanks, Frida. I have some questions for Aoife. Is there data on the percentage of cases from dining out? People are asking.
So we I had mentioned on my slide from where that information is available. We collected that from 2012 onwards, where we have information on that. 64% of cases had reported that they dined out in the 3 days prior to their symptom onset. I don't know if there's any more to that. Does that answer the question? I had maybe wasn't clear from the slides. We can provide a yearly breakdown as well, if that would be of interest to people.
Okay, we can get back to the person that asked the question and ask them if they want that. They've also asked what HPSC identified as source of the prevalence of cases of Salmonella in a 4-year-old children. Sorry.
In the 0 to 4s. Again, probably multifactorial thing going on there, in that children, I suppose, are less, well able to keep up with their own personal hygiene in terms of toileting and hand washing, particularly before they handle food or after handling pets or being out outside, playing in mud or whatever, maybe less likely to wash their hands. I think the other thing is the disease can sometimes be more severe in that age group, so they're more likely to be tested. While milder infection in an adult, because it's self-limiting, they may not go to their GP or get tested and confirm that they've had Salmonella, where I think with the child, if they have that more severe diarrhea, particularly bloody diarrhea, a parent will be worried enough to get them tested. So I think a few things going on there. The spread among children is probably more common, and then also the disease is quite severe in that group.
Okay, thanks, Aoife. I have another question for Frida here. They say, I know we take action in breeders, but in broilers and poultry products, there has been an increase in this CRVR for sure. Okay, maybe that's someone answering something, is it? Maybe I'm getting comments instead of questions.
I think we've already discussed that. I think that had to do with the Infantis thing.
Apologies. Okay, a question for Gary. Has Moy Park considered AMR testing in the future for Salmonellas in particular?
Yes, everything's open for looking at, but I think with AMR testing, one of the things we need to consider is actually, if you do get a result back, is understanding how do you interpret that result, where does it come from? Because obviously AMR can be natural in the environment, and you can't pick up AMR from medication that birds have never seen. So again, it's trying to understand that. So yes, AMR testing will be considered and is being considered, but it says it's just the interpretation of that data and the results that come back. Make sure that you're looking at the right thing and not chasing a red herring completely.
Perfect, thanks, Gary. And I have another question for you on vaccination.
Yeah.
SE and ST vaccination are permitted in the UK. Are they used by Moy Park, and what are the considerations?
The only considerations really is if some countries we're exporting to don't want it, so that's something we need to consider, that if you're depending on your export market. Within the UK, we do it, but within some export markets, if we were planning to go to a certain country, and again, off the top of my head, I don't know which ones they are, but they are the ones that you would need to consider. You couldn't vaccinate for certain flocks for that.
Perfect, thank you, Gary. I think that's the end of our questions. Am I right, Tracy? Just in case I've missed. There was a lot.
No, that's it, all answered.
Yeah,
thanks,
perfect.
Mairead. Thanks to everyone for that.
Thanks everyone, and thanks everyone for joining us. I suppose just to finish off, if you haven't joined our knowledge network, you can do so at safefoodkn.net, and that'll keep you up to date on all our resources and upcoming events. And also you can follow us on LinkedIn for that. We'll also be sending out an email shortly with the recording of this webinar and an evaluation form, which we'd really appreciate if you could take a few minutes to fill out. And I'd like to thank all our presenters again for taking time out to join us and answering all the questions so thoroughly. Thank you very much. Anything else, Tracy?
That's it. No, thank you, everyone. Thanks for joining. Thanks to